Hello everyone. In this tutorial, we are going to talk about recombinant DNA technology, or we can also call it genetic engineering or gene cloning, its applications in the fields of agriculture. Before we get into the topic, the first and foremost thing which we should understand is what exactly is recombinant DNA. Recombinant DNA are molecules of DNA from two different species that are joined together and are inserted into a host organism to produce proteins that are of value to science, medicine, agriculture, and industry. Recombinant DNA technology relates to the usage of three main tools. The first tool is enzymes. Enzymes like restriction enzymes, polymerases, and ligases. The restriction enzyme plays an important role in this technology. It cuts at a specific site within the DNA molecule called a restriction site. Usually, the restriction enzyme will produce sticky ends in the DNA sequence that will help it bind specifically to the desired gene. Other enzymes like polymerases helps in synthesizing DNA, and ligases helps in joining two DNA strands. The second tool, vectors. Vectors are vehicles that carry gene of interest into the host organism. Several types of vectors have been developed. The most commonly used vectors are plasmid and bacteriophages. A vector must contain the same restriction site that are found within the desired gene for facilitating gene integration. Besides, vectors should also carry sequences for selection and identification, such as a gene for antibiotic resistance and cloning site. The third tool, host organism. The host organism is the cell in which the recombinant DNA is introduced. Host organism could be bacteria, fungi, or animal cell. To introduce vectors into hosts, technique involving microinjection, ballistic technique, gene gun, alternate cooling and heating, calcium phosphate ions have been employed. To get a clearer picture, let's look at a simple animation. A bacterial cell has single circular molecule of DNA called the bacterial chromosome. It also has a small extra chromosomal DNA molecule which replicate independently known as plasmid. These plasmids are a suitable vector or carrier that can carry our gene of interest inside the host cell. The DNA or gene of interest is isolated from the host cell, inserted into the plasmid, which is possible with the help of restriction enzyme, also known as molecular scissors, producing sticky end. The gene of interest incorporates within the plasmid with the help of this sticky end. The gap between these two strands were sealed using an enzyme known as ligase, or also called as molecular glue. The recombinant DNA is now ready to be inserted inside the host cell to produce the desired product. Recombinant DNA technology or genetic engineering have been largely used in agriculture for improvement of crop yield, resistant to pests and herbicide, which were also known as genetically modified crop. First one, we have flavor saver tomatoes. The flavor saver tomato was introduced as the first genetically engineered whole food in 1994. The tomato was produced by inactivation of polygalactoironase enzyme. Non-availability of this enzyme prevents over-ripening because the enzyme is essential for degradation of cell wall. In the picture, we can see ordinary tomatoes with the flavor saver one. On the left hand corner we have ordinary tomatoes, and on the right hand corner we have two rows of flavor saver tomato. After 10 days of post harvest, all tomatoes look equally or healthy, and after 20 days the ordinary tomatoes looks a bit soft, their skin begin to shrink, whereas no change was observed in the flavor saver one. After 45 days, ordinary tomatoes began to rot and became unfit for consumption, whereas the flavor saver one, there was not much change. So this clearly shows the silencing of polygalactoironase gene using genetic engineering helps in minimizing post-harvest crop loss. The next genetically engineered crop is the famous BT crop. Bt comes from the soil bacterium Bacillus thuringiensis, from which Bt toxin are taken. The toxin is insect group specific and is coded by a gene named Cry. Some species of plants like corn, sweet corn, brinjal, cotton, etc. are engineered to produce their own Bt. This is done by inserting Cry gene into them from Bacillus thuringiensis. Plants use this DNA to produce their toxin Bt protein in their tissues. When insect larvae feeds on the plants, they are exposed to this toxin and they die. For insect, the Cry protein is harmful because it changes its shape inside insect alimentary canal and become toxic, whereas for humans, it does not change its shape. Thus, it is not harmful. This technique also reduces spraying of pesticide to control insect. The next genetically engineered crop is golden rice. Many people in developing countries are vitamin A deficient, meaning they don't receive enough vitamin A in their diets. Vitamin A deficiency is a serious public health issue in parts of Asia and is a leading cause of childhood blindness because rice is a staple food for millions of people in countries with high rates of vitamin A deficiency. Many scientists and humanitarians believe golden rice could be a novel solution to vitamin A deficiency. Rice breeders have been working to incorporate the golden rice trait in rice varieties from these countries. Golden rice is a transgenic variety of rice, Oryza sativa, that contain high levels of beta-carotene, the precursor to vitamin A. It was first developed by two academic scientists in Europe in the 1990s and has attracted significant attention and controversy since then. Golden rice gets its name because the high levels of beta-carotene makes it a golden yellow color, which helps in curing vitamin A deficiency diseases like night blindness. The Philippines is moving closer to approving golden rice varieties for cultivation, and Bangladesh has announced it is likely to approve commercial production in 2019.